So cyanobacteria are also called as blue-green algae. So which is the smallest living cell that is present on earth? It is Microplasma, thermophiles. So these bacteria, they survive in hot spring and they can also survive in deserts, they can also survive in snow, and also they can survive in deep ocean. Methanogens, they are the bacteria which are present in the gut of the ruminant animals. Hello everyone, a warm welcome to today's session on chapter 2, that is biological classification. I am Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session, we had discussed about the 5 kingdom system of classification, which is also called as the Whittaker system of classification. So in that session, we had dealt with the 5 kingdoms that was proposed by Whittaker, which is Kingdom Monera, Kingdom Protista, Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia. And we also discussed what drawbacks in Linnaeus 2 kingdom system of classification led to the Whittaker system of classification or 5 kingdom system of classification. So in today's session, we shall discuss about the first kingdom of classification given by Whittaker, that is Monera. So we shall discuss about Kingdom Monera. So bacteria are the sole organisms that are put under Kingdom Monera, and we know that there are thousands to lakhs together of bacteria that are present in our surroundings and are living with us. So these bacteria, they have some characteristic features in them. So what are they? We shall study. So bacteria, they usually present in soil, that is we call it as the habitat of bacteria. Habitat means the area or the region or the place in which an organism lives. So what is the habitat of the bacteria, or where do the bacteria live? They live in soil and they live in hot springs. So can you imagine, in hot springs, they are very, very hot. They have water in the form of springs which are very, very hot. So very hot, we cannot even imagine that any organism can survive. But there are certain bacteria which are called as thermophiles. So these bacteria, they survive in hot spring. And they can also survive in deserts, they can also survive in snow, and also they can survive in deep ocean. So in deep ocean, it is difficult for the organisms to survive because there is no sunlight, there is no feeding source, that is, there is no food source, because plants also do not grow. But since bacteria are saprophytes, they can grow on dead organic matter, they even survive in the deep dark ocean. So this is the diverse habitat that the bacteria has, and that is why they have been placed under a separate kingdom called as Monera. And when you look at the structure of the bacteria, it is very simple. Why it is simple? Because it is unicellular. It consists of only one single cell. Unlike plants or animals, they do not have multiple cells. They are not made up of a large number of cells, which leads to complications, or it does not form a complex system. Therefore, they are very simple. The structure of bacteria, since they are unicellular, it is very, very simple. But when you consider the behavior of bacteria, it is very complex. Why it is complex? Because unlike we human beings, bacteria, they cannot move from place to place because of the environmental changes or environmental factors or whatnot. But in spite of any of the environmental changes or issues that is there, the bacteria tend to thrive or survive in that particular situation. Therefore, the structure, though it is very simple, the functioning of the bacteria or the characteristics of the bacteria or the behavior of the bacteria is very, very different. It is very complex. And talking about the metabolic diversity. So metabolic diversity, metabolism is something related to food, the digestion of food, and the digested food being utilized by all the other parts of the body for proper functioning. So how does the metabolic activity or metabolism take place in bacteria? In bacteria, they are autotrophic. So auto means self. So they can synthesize their own food on their own. They can be autotrophic, wherein they can be photosynthetic autotrophic. So photosynthetic autotrophs means the best example is the blue-green algae, that is the cyanobacteria. So cyanobacteria, they have chlorophyll pigments in them. They have pigments which give them the green color. That is why they are also called as the blue-green algae. But though it is called as the blue-green algae, it is put under bacteria because it is a bacteria. That is why cyanobacteria, it is also called as cyanobacteria. So these cyanobacteria can synthesize their own food. They are autotrophic, and so they can also be, bacteria can also be chemosynthetic autotrophic. So chemosynthetic autotrophic means they can depend on the other organic substances that are present in the soil for their food source, because I told you bacteria also live in the soil. Next is they can be heterotrophic, that is they can completely depend on other organisms for their food. So in detail we shall study what is photosynthetic, how the bacteria can be photosynthetic autotrophic, chemosynthetic autotrophic, and how they can be heterotrophic. So we shall study about these in detail later. Before that, we shall study the different groups of organisms or different groups of bacteria that are placed under the kingdom Monera. So before moving into that, we shall study the different structures of the bacteria or the shape of the bacteria. So the bacteria can be round shape, which is also called as cocci, can be oval or round shaped, that is it is called as cocci. They can be oval or round shape, it is called cocci, and they can be comma shaped, that is why they are called as vibrio, that is they can be like commas. They can look like commas, that is why they are called as vibrio bacteria. They can be cocci, that is round shape or oval shape or circular in nature. They can be in comma shape, that is why they are called as vibrio bacteria, vibrio bacteriae, and they can be rod shape. They can be like this rod shape. It can be like a rod, or bacilli shaped, rod or bacilli shaped, and they can also be spiral shaped. So just like a spiral, they can be spiral shaped like this, spiral. So these are the different shapes in which the bacteria is present. They can be rod shaped, they can be cocci, they can be vibrio, comma shaped, and they can be spiral shaped. So after studying all these, that is the habitat of the bacteria, the structure of the bacteria. So based on the structure, they can be of different shapes, and based on the metabolic diversity, they can be autotrophic, wherein they can be photosynthetic or chemosynthetic, and they can also be heterotrophic. So under bacteria, different groups are there. That is under the kingdom Monera, there are different groups. So we shall study about them one by one. So the first one is Erybacteria. So Erybacteria, they have a peculiar habitat. So those bacteria that live in areas where there is too much of salt, or those bacteria that live in extreme salty areas, come put under the Erybacteria. So you should understand with this, as I told you, the bacteria, they have a diverse habitat. So some bacteria can live in extreme conditions of the earth. That is, they can live in high salty areas. They can live in high temperature, where the temperature is very, very high. The best example is they can live in hot springs, and also they can live in marshy areas. You know that marshy areas, the water consists of a lot of chemical compounds, harsh chemical compounds, because marshy areas, always there will be dead and decaying matter to present there too much. That is why. So habitat, so those bacteria, now you can guess. I'm telling you, Erybacteria, they have a habitat, or they live in extremely salty areas, they live in hot springs, and they also live in marshy areas. So what it means, those bacteria which live in extremely harsh areas are grouped under Erybacteria. So those bacteria, usually they are of three types. They are halophiles, they are thermoacidophiles, and they are methanogens. So Halophiles. What are halophiles? Halophiles are the bacteria which are grouped under Erybacteria, and they can live in extremely high salt conditions or salty areas. And what are thermoacidophiles? Thermoacidophiles are the bacteria. So thermo, something related to temperature. Acido, something related to acidic nature. So hot springs, they usually have high temperature and high acidic nature in that particular area. So the bacteria is able to withstand or live in hot springs. So such bacteria are called as thermoacidophiles. And some bacteria can live in marshy area, be it in the swamps or be it in the gut of the animal. So gut of the animals, that is the intestine region of the animal, is also slimy and marshy, right? In that particular region also they can live, and they are such bacteria which live in marshy areas are called as methanogens. Why they are called as methanogens? Because we shall see. So now you can question, how are the Erybacteria able to survive in extreme conditions? It is because of the cell wall structure. So the bacteria of the cell wall, it is made up of polysaccharides and amino acids. We have seen in the previous session. And these cell wall structure, the cell walls are very, very rigid and thick, and they are able to survive in the extreme salty conditions, in the hot spring, as well as in the marshy conditions. And I told you about methanogens. So why methanogens, why the bacteria which live in marshy areas are called as methanogens? They can be called as something else, right? Why do they give the name methanogens? Why? Because methanogens, they are the bacteria which are present in the gut of the ruminant animals. So ruminant animals, what are ruminant animals? The animals which feed on fodder, that is especially fodder such as, or which produce methane. So you know that cow and buffaloes and all, they release methane. So when they feed on the grass, what the grass will get digested and lot of methane gas will be released, and their gut is filled with methane. It is a methane environment in their gut. So that is why in ruminant animals. So what are ruminant animals? Those animals which feed on fodder, therefore which convert their food into methane in their gut. The best example is buffalo, cow, all that are ruminant animals. So these ruminant animals, they produce methane gas in their gut or in their intestine or in their stomach. And the bacteria are able to survive the methane. You know that methane is very, very poisonous, right? Even if if we inhale the methane gas, it is hazardous to the human health. But imagine the bacteria, because of their rigid cell wall structure, they are able to live in the methane environment of the gut of the ruminant animal. So that is why they are called as methanogens. So methanogens, they are the bacteria that live in the gut of the ruminant animals where methane is present. So these are methanogens. And also these bacteria can also survive in the dung. The dung contains methane, and these bacteria, they can also survive in the dung of the animal. So therefore they are called as methanogens. So this is about Archaebacteria. So those bacteria which can live in extreme conditions such as the salty areas, hot springs, and the marshy areas filled with methane, such bacteria are grouped under Archaebacteria. So next, moving on to Eubacteria. So these bacteria are also called as true bacteria. Eubacteria are also called as true bacteria. So these Eubacteria, they have a very tough or a rigid cell wall. They have a rigid cell wall and they can move. So in Eubacteria, again, some are motile, some are non-motile. Motility means the ability to move from one region to another. So some bacteria under Eubacteria, they can move, some cannot move. But can you see here in Archaebacteria, there was nowhere we had mentioned about the motility. These are something different. The bacteria which can live in extreme conditions were all placed under Archaebacteria. But Eubacteria, they can move. Some of the eubacteria, they have flagellum and therefore they can move. They are motile and they have a vast metabolism diversity. So what are the metabolism diversity in them? They can be photosynthetic autotrophs, they can be chemosynthetic autotrophs, and they can be heterotrophs. So we shall see. So there, those bacteria, Archaebacteria and Eubacteria, now you know they have, again in Monera itself, they classified Monera into two different sections, that is Archaebacteria and Eubacteria. So this is classification, how you minimize the number of organisms under one particular group based on the characters. Again and again you divide the organisms. So under Eubacteria, they can be photosynthetic autotrophs, they can be chemosynthetic autotrophs, and they can be heterotrophs. So first we shall study about the photosynthetic autotroph. So I gave you the example of Cyanobacteria. So Cyanobacteria are also called as blue-green algae. So why blue-green algae? Because they have chlorophyll pigments in them. So what do the chlorophyll pigments do? Even plants have chlorophyll pigments. So you can ask me, even plants have chlorophyll pigment. They could have put Cyanobacteria, which is also called as blue-green algae, under plants, right? But no. Why? Because there is some, they are unicellular. That is why. But plants are multicellular, right? So that is why they have been grouped under bacteria, which are unicellular. They have been put under prokaryotes. That is the reason. So Cyanobacteria, they are the photosynthetic autotrophs. Why? Because they have chlorophyll pigments in them and they are unicellular. But what happens here is, every time students, they get confused thinking that they are multicellular. You can ask me now, ma'am, they are multicellular, right? No, they are unicellular, but each cell, they occur in groups. They occur in separate groups. And they look like multicellular, but they are unicellular. These are separate, they are unicellular. So they are unicellular, that is, they can occur in colonies. So each cell here, it can be considered to be a sign. So they can be unicellular and they occur in colonies, that is, either they can be filamentous, they can be filament. So this is filamentous, they can be filamentous. They can occur in groups or in colonies, and also these cyanobacteria, they live in fresh water. They can also live in marine water, that is sea water or ocean water, and they can also live in soils, that is, they can be terrestrial. So what is the body structure or cellular organization of cyanobacteria? They are unicellular. Though they are unicellular, they occur in colonies or they can occur as filaments, and they live in, where do they live? What is the habitat look like? They live in fresh water. They can also live in marine water, that is oceans and seas, and they can also be terrestrial, that is, they can live in soil, they can live on land. So this is the habitat. And the colonies, so I told you, though they are unicellular, they occur in colonies. They occur in colonies like this. And these colonies are covered by a mucilaginous sheath. So colony one, second one, they all group together to form colonies. And once they form a colony, they are covered by a mucilaginous sheath. So this is the mucilaginous sheath that covers them. So this thing is the mucilaginous sheath. So they are covered by a mucilaginous sheath. And one more thing is, you have to know that under cyanobacteria, they are under eubacteria. They are one example for eubacteria, and these cyanobacteria are photosynthetic autotrophs. So those eubacteria, and they can be photosynthetic autotrophs, they can be chemosynthetic autotrophs, and they can also be heterotrophs. So those, some of the bacteria under eubacteria are photosynthetic autotrophs, and the best example is cyanobacteria, which is also called as blue-green algae. Cyanobacteria, they have both advantage as well as disadvantage. They are useful as well as they are not useful to the environment. What is the main disadvantages? They cause algal blooms. So what are algal blooms means? Like you might have seen in the rivers, there will be a mat. I'm not talking about azolla. Azolla is a plant. I'm not talking about that. But otherwise, you can see algal blooms wherein a complete green mat will be covered on the riverbed. So what happens if the green mat, that is nothing but cyanobacteria. Algal blooms also will be covered. That is, azolla will also cover. But I'm not talking about the plants. I'm talking about the cyanobacteria here. These bacteria will form a green mat. So when a green mat is formed, what happens? The light will not be able to penetrate. So if the light doesn't penetrate, green plants will not grow in a particular river, right? So if green plants don't grow, how will the fishes feed? It is difficult for them to feed and survive. It will cause death of the fish, and not just that, it will also pollute the river or the ponds. So therefore, it is hazardous to the life of the animals that are living in the river. That is nothing but algal blooms. Algal blooms are nothing but they completely cover the entire upper surface layer of the water or the river or the pond. So that is one disadvantage of cyanobacteria. But apart from that, these cyanobacteria can be used as fertilizers. So on the river, whatever is growing, it can be collected and can be used as fertilizers for the crop. Why? Because cyanobacteria, they help in fixing atmospheric nitrogen. And you know that in plants, nitrogen is very, very important. It is one of the main nutrients. It is one of the macronutrient that is required for the plant for its good growth, right? And plants cannot fix their nitrogen. They depend on other organisms to fix the nitrogen. And these cyanobacteria, they help in fixing the nitrogen. Therefore, it acts like a fertilizer to the crop. So cyanobacteria, they can be added to the crop fields for that. So they help in fixing atmospheric nitrogen. So there is one cell present in cyanobacteria. It is called as heterocyst. So this heterocyst is the one that helps in fixing the atmospheric nitrogen. The best example for cyanobacteria is the nostoc and Anabaena. So nostoc and Anabaena are put under eubacteria, especially under photosynthetic autotrophic eubacteria, wherein they are nothing but two types of cyanobacteria, that is nostoc and Anabaena. So this is the example for cyanobacteria. So next, moving on to chemosynthetic autotrophs. So chemo means something related to chemicals, synthetic, synthesize. So they are the bacteria which oxidize the inorganic substances that are present in the soil, such as the nitrates, and the ammonia, and whatever energy is released. I told you they help in fixing or atmospheric nitrogen, right? That is different. But those are photosynthetic autotrophs, okay? They are different. But here, chemosynthetic autotrophs are the bacteria. So bacteria, I told you, they also terrestrial. That is, they grow in the soil. So whatever inorganic substances that are present in the soil, such as in the soil we have the nitrates, we have the nitrites, the ammonia, all that is present in the soil, right? So they oxidize these inorganic substances, and whatever energy is released during the oxidative process, that they will use for the ATP production, and that is how they produce their food. And also they help in playing a very important role in recycling the nutrients that are present in the soil. So you know that a little bit of energy from where it is taken, it will be given back. So again here what happens during this process, again some of the nitrates or the nitrites or the ammonia are given back to the soils, especially. So therefore they help in recycling the nutrients that are present. So during this process of oxidation of inorganic substances into ATP, that is adenosine triphosphates, what happens is there is recycling of the nutrients. The soil, they in turn utilize the nitrates, nitrites and ammonia that is present in the soil as a food source, and then again they give it back to the soil, wherein they increase the nutrients present in the soil, such as the nitrogen, phosphorus, the iron and the sulfur. Therefore, they make the soil very fertile. So this is one advantage of the chemosynthetic autotroph. So they make the soil very fertile, wherein they give the soil nutrients like nitrogen, phosphorus, iron and sulfur. So this is one best advantage of the chemosynthetic autotroph. So next, moving on to the last one, that is the heterotrophic bacteria. So I told you, eubacteria, they can be chemosynthetic autotrophs, they can be photosynthetic autotrophs, and they can be heterotrophic. So heterotrophic, hetero, they depend on other organisms for their food source. So they are the important decomposers. So how do you think whatever dead plant or dead animal that is present in the land, how do they decompose? Will they decompose as such if you put it in the soil? No, it is because of the presence of the bacteria in the soil, they get decomposed. So these heterotrophic bacteria, they are the important decomposers. So they decompose the dead organic material and then they use that dead organic material as their food. That is why they are called as heterotrophs. Means they're depending on other organisms. They are eating the dead organic matter. They are feeding on the dead organic matter. And apart from that, these heterotrophic bacteria, they are of various use to the human beings. What are the uses? We shall see. They help in making curd from milk. So lactobacillus, you have the best example for one bacteria, that is lactobacillus. It helps in the curdling of the milk. The curds that you get, how it gets converted into curd, it is because of the action of a particular bacteria called as lactobacillus, which is a heterotrophic bacteria. So therefore, heterotrophic bacteria, they help in making curd from milk and they help in production of antibiotics. The various antibiotics that we get in the market, it is all got from heterotrophic bacteria. Then they help in fixing nitrogen. The best example, we have seen how bacteria, they help in fixing nitrogen. Then they help, the best example is the rhizobacter or the rhizobium, which helps in fixing the atmospheric nitrogen, especially in the leguminous roots. So leguminous roots, what are the leguminous plants? All the bean families, bean plants, they are all leguminous. So if you pull out a bean plant, you can see in the root, there will be nodules present. So in the root of the bean plant, you can see nodules being present like this. So in these nodules, the rhizobium or the rhizobacter is actually present, the rhizobium or the rhizobacteria. So these rhizobacter or the rhizobium, they help in fixing atmospheric nitrogen, because what did I tell you? Plants cannot utilize nitrogen as such. They take the help of the bacteria to fix the atmospheric nitrogen. Therefore, these heterotrophic bacteria which are present in the soil, one of the best example is the rhizobacter or the rhizobium. They help in fixing atmospheric nitrogen in the leguminous roots. And apart from this, these heterotrophic bacteria, they can be disadvantageous or harmful also. How? They are pathogenic. What are pathogens? Those organisms that cause diseases are called as pathogens. How they are disadvantageous is because they cause harm to human beings, they cause diseases in crops, they cause diseases in farm animals and also to pet animals. So we human beings also, we are subjected to a lot of disease because of various bacteria that are there, right? So they cause diseases. That is why they can be pathogenic as well. They can have various advantages and various disadvantages also. So they cause diseases such as cholera, typhoid, tetanus in human beings, and in plants they cause citrus canker. So plant disease caused by bacteria is citrus canker. Your diseases caused in humans or animals, it is cholera, typhoid and tetanus. So therefore they can be useful as well as they can be harmful to the human beings. So next, let's study about how do these bacteria, that is the organisms that are present under the kingdom Monera, they reproduce. So bacteria usually reproduce by fission. What is fission? Splitting. You're dividing the cells. Here what happens here is, you can see there, this is the bacteria and then it has the genetic material. So it has the genetic material. So the DNA, that is the genetic material. And I told you they lack a nuclear membrane. So imagine if they have a nuclear membrane, you'll have the nucleus, then in the nucleus the chromosomes will be present and in this the genetic material will be present. But bacteria, they do not have a nuclear membrane. They don't have a nuclear membrane. But instead, that is why they are They don't have a true nucleus, but they have the genetic material. So this, when the bacteria splits, the genetic material moves to also splits, and the two bacteria cells are formed separately. So this is nothing but fission, splitting. Splitting will take place. So one bacteria, they will split or divide into two cells, and each cell will have their own genetic material, that is the DNA, and they form a separate. Now again, these will split, they'll form four bacteria. That is, they reproduce by fission. That is asexual kind of reproduction, wherein they increase their number. They multiply. So they reproduce by fission. And also, bacteria, they can reproduce asexually by producing spores. There are some bacteria, during unfavorable conditions or during extreme conditions, they start producing spores. Instead of reproduction by fission, they undergo a type of asexual reproduction, wherein they start to produce spores. And also, they can reproduce by sexual reproduction. Sexual reproduction is nothing but here, wherein they undergo fission, that is, transfer of DNA will take place. They also reproduce by sexual reproduction, wherein transfer of DNA from one bacteria to other will take place. So bacteria, they can reproduce asexually, that is by producing spores, and they can also reproduce sexually by transferring the genetic material, that is the DNA, from one bacterium to another. And the mode of reproduction in bacteria mainly is by fission, that is, they divide, the cells divide and form new daughter cells. The parent cell will form new daughter cells. So this is about reproduction. So under Eubacteria, there is one more group of organism that has been put under the kingdom Monera, that is the Mycoplasma. So Mycoplasma, they are the smallest known living cells that is present till date. So which is the smallest organism that is present? That is Mycoplasma. Mycoplasma, they do not have a cell wall. So the cell of the Mycoplasma, it is not covered by any cell wall, which means they completely lack a cell wall. And also they have a peculiar feature wherein they can survive without oxygen. So the Mycoplasmas, they can survive without oxygen, and they have a disadvantage, that is they can be pathogenic to animals and plants. So the best example here I've shown in this picture, wherein you can see Mycoplasma, they usually infect the lungs of human beings. They cause diseases in the lungs of the human beings. So therefore, they are pathogenic to not just the animals but also to the plants, wherein they cause a lot of diseases in the plants as well. So this was about today's session, wherein you studied about the kingdom Monera, wherein under the kingdom Monera, we studied about two groups of bacteria, that is Archaebacteria and Eubacteria, and we also studied about Mycoplasm in separate. So in the next coming session, we shall move to the second kingdom classified by Whittaker, that is Protista. So we shall study about kingdom Protista in the next coming sessions. I hope you have understood the session well. We shall meet again in the next session. Thank you.